Showtime!
Ladies and gentlemen, please make your way to your seats.
Showtime!
Please make your way to your seats. The show is about to begin.
Showtime!
Let the magic begin.
Hacking DNA to make next-gen semiconductor materials. I felt like we've talked about this already, but I think they're doing this more and more and more. So what's going on with this one?
Yeah, this is the thing. I had to reread and because like I post articles and then we talk about them like weeks, months later, right? So I definitely had to go back and I was second guessing myself as if we talked about this before, but we didn't. So this again, like you said, it's constant research is happening, advancements are happening. So new articles are being posted all the time. So in this one, researchers have developed a universal method to produce a variety of complex 3D metallic and semiconductor nanostructures using DNA as a scaffold. Now, just for those who haven't heard the episode before, DNA's programmability allows it to self-assemble into precise arrangements. And so the researchers were able to use vapor deposition and liquid infiltration to coat the DNA templates with inorganic materials like silicon, zinc oxide, and platinum. So the combination of DNA-directed assembly and material templating has enabled efficient exploration of vast material design space to identify promising structures for advanced applications. Now, just to give you some more information here, we did talk about nanotechnology and DNA. This has evolved tremendously over the past decade. So the challenge has been transferring structures to robust functional materials, and this is kind of, I think, the next step. So the vapor liquid infiltration of inorganic precursors into the DNA template, that helps to overcome the limitations of purely organic structures. So the multi-step process, what it does is essentially creates like a backbone while preserving the underlying architecture, which is really, really cool. Just to touch on it quickly, metallic semiconductor and oxide nanostructures, they have applications in electronics, photonics, energy conversion and storage. We can get into that in more detail. But just to give you a little bit more background, DNA itself, for those that don't know, it has a programmable base pairing. What that does is it enables it to fold into highly ordered 2D and 3D structures like tiles, lattices, and even origami. So self-assembly, it's driven by hydrogen bonding between two complementary DNA sequences. So strand design itself, it determines the final configuration. The thing with DNA nanotechnology is scaffolds can be tuned for precise shape, rigidity, and porosity, which allows them to bond it with all this other stuff. So there's a lot of advanced technology that's being used in this, robust chemistry, modeling software, sequencing tools, a whole bunch of different things that are enabling this, which is really, really cool.
Yeah, in the past, we've talked about self-replicating DNA robots, and we talked about gene editing, how AI is creating DNA blueprints, and we've talked about harnessing DNA for next-gen data storage. Yeah. And we've talked about 2D and 3D structures, but they were mostly RNA-based, I believe, if I remember. So can you explain how is this DNA nanotech being used in the field of medical nanotech? Like, I'm always great. You can do stuff really at this micro level now, but what are you doing with it?
Yeah. Why? Like, what's the point of it? Right. So a couple things. So I know I touched on DNA origami. So drug delivery, right? So DNA origami and DNA coated nanoparticles can encapsulate and selectively deliver drugs to targets like tumors. So the DNA guides binding to cancer cell surface receptors for precision targeting, which is really interesting. Again, biosensors. So I know we just touched on it with borofine, but DNA can be designed to bind specific biomarker proteins which will enable highly sensitive detection of disease markers, which is really cool. And medical imaging. So DNA nanostructures with high cellular uptake, they can incorporate contrast agents for MRI and fluorescent imaging, which enables non-invasive visualization of biological processes. This is controversy, but vaccines. So the repetitive surface patterns of DNA tile lattices, what they can do is they can display viral antigens to provoke stronger immune responses for vaccine development, which is really cool. We've got just talked about cancer therapeutics, tissue engineering. So DNA-based hydrogels could provide scaffolds for cell growth and organization, which it can be tunable to the stiffness to promote tissue regeneration. So the cool thing is it's biocompatible. It's nanoscale, it's programmable, and it's got a precise molecular recognition of DNA, which makes it an ideal material for advancing medical nanotechnologies.
And on the article here, it says they're already using their technique for other applications like improving microelectronic materials or gas separation membranes for hydrogen.
Yeah, which is really cool. I don't know what that application means. Maybe it's, but yeah, it's really interesting to see. This is where. It's not just kind of theoretical. This is where they're a little bit further ahead. They've actually bound these biomaterials together, and now they're doing testing and different things like that, which is really cool.
Yes, this universal method was extremely effective for producing 3D nanostructures of a wide types of material to such an extent that it surprised researchers. This says the team was able to produce 3D nanostructures containing different combination of zinc, aluminum, copper, molybdenum, tungsten, iridium, tin, platinum. And this is the first demonstration of its kind for creating highly structured 3D nanomaterials in a manner that's straightforward, repeatable, and robust.
Yeah. And the next thing is the surprising thing is that they were able to successfully produce so many different material compositions. So it's showing that it's repeatable, which is key to this, right?
Yeah, straightforward, repeatable, and robust.
Yeah.
To prove the success of this method of each nanostructure, they developed the researchers leveraged expertise in world-class imaging facilities. Brookhaven Labs who produce ultra-bright X-rays to illuminate the physical, chemical, and electronic makeup of the samples at the atomic scale. Like this is one of those things too, is it because the things are at so small, you've got to rely on these types of things to validate it. Otherwise, yeah.
Exactly.
So,
yeah. How do you validate it without developing it and testing it in human tissue or human body, right? So there are ways, and they're being, so that's the other thing too, is they're coming up with innovative ways to prove this technology too.
So what are the challenges of taking this to mass production and start using them in nanotechnology and medical labs?
Yeah. So immune responses. So DNA structures potentially could trigger unwanted immune responses in the body. So they're going to have to come up with strategies to cloak the DNA against immune detection. So that apparently is being developed. DNA nanostructures could bind unintended cellular targets, leading to toxicity, which is scary. So they're going to need to do more research to understand biodistribution. Again, like everything, scale up production. So mass producing complex DNA nanostructures is still very challenging and expensive. So they're going to have to improve synthesis and purification methods. Stability. So the DNA nanostructures can be degraded in biological environments. So they're going to have to come up with chemical modifications and coating. Regulatory approval, clinical tests, in vivo testing. So many DNA nanostructure systems have only been tested in vitro. So they're going to have more assessment in relevant biological environments. So there's a lot of advantages in this, and the field still has challenges to overcome for practical use, medical use of DNA nanotechnology. But like we've covered, like we've only been doing these episodes for a little bit, and there's already advancements happening, which is really cool.
Yeah, I mean that's what I'm saying. We see this and like, wow, it's moving fast.
Yeah.
And I love to see what they're doing here. And I think, this group previously used this technique to reveal the 3D structure of DNA lattices with single particle resolution. And that was this X-ray technique they were using to check the resolution. How close? Like, are we going to, like, tomorrow, 5 years, 10 years?
So in the near term, we're talking 1 to 3 years. I would say specialized research tools and kits will continue gaining adoption. So the more people will be using, will be getting into the research and doing research. If they partner with biopharma, that they're going to need more investment for proof of concept studies and different things like that. 3 to 5 years out, that's when they'll probably get into the scalable manufacturing methods to bring down the costs. And then advancements will allow for customizations for specific applications. And obviously, we'll probably get into some regulatory review and different things like that. Long term, 5 to 10 years, potentially we'll start seeing precision medical applications. But again, it probably will be very expensive still. But if the regulatory gets approved and tests go well and clinical trials go well, there could be more mainstream adoption, like in molecular diagnostics, biomanufacturing, even data storage and optics. So DNA nanotech could be a commonplace within the next decade. Again, 10 years out, I really want to do like a time capsule and see how far away we are.
Wow, that was